I am fairly new to elderberry. I started within the last year, but so this project or this presentation is going to mostly focus on a little brief introduction on what's been done, and then I'll move into our goals for elderberry breeding projects. American elderberry, or Sambucus canadensis, is native to Eastern and Central North America, and it has been used extensively by indigenous people, and often in a medicinal capacity. And as of 2017, there were nearly 500 producing acres and almost 800 planted in the United States, and over 40% of this acreage is in Missouri. And then there's a few pictures there of current uses of elderberry. So American elderberry is a medium to large bush or small tree. It puts out a lot of suckers, prefers moist, well-drained sites with plenty of sun, and is hardy to zones 4 through 10. And then in the Midwest, it produces clusters of flowers in June that mature to dark purple berries in late summer, so around August. What we know so far about genetics and reproduction, there is some amount of self-incompatibility in elderberries. So a given cultivar can't pollinate itself necessarily. So it's outcrossing, and this is why it's often recommended to plant multiple cultivars together in an orchard. It is likely a diploid, so 2 sets of chromosomes with 36 total chromosomes. And on this side of the slide here, there's a diagram showing genome sizes. So a 2012 estimate of American elderberry put the genome size at about 14 billion base pairs. And then our work, we sent some tissue to Oregon State, and they estimated about just over 12 billion base pairs for our tissue. European elderberry, just over 13 billion. And then for comparison, the human genome is just over 3 billion. So the American elderberry and European elderberry, very large, complex genomes. So more recent organized breeding and selection efforts have mostly been in New York, starting in the 1920s and 30s, with the release of Adams 1 and 2, Ezioff, and York. And then there have been several others from the Nova Scotia Experiment Station in the 50s and 60s. And then more recently, there have been a lot of selections from the wild around the Midwest that were trialed and released through the efforts of people like Andy Thomas, Pat Byers, Terry, and other collaborators. So our group's breeding and research goals, we plan to try to build on current progress to develop improved cultivars. We will perform targeted pollinations to further our understanding of cross-compatibility, and that plays into that self-incompatibility, and develop populations. And then we'd like to do a more in-depth characterization of flowering for popular cultivars and perform a reference genome assembly. So each of those things that I mentioned supports our breeding efforts. So phenology, or flower development, helps us to understand the compatibility of timing between different cultivars. Cross-compatibility helps us understand self-incompatibility and which genotypes or cultivars can be crossed together successfully. The reference genome will lead to more genomic resources that we can use to help us develop improved cultivars. And then, of course, the interdisciplinary collaboration and grower input will inform our future goals and strategies.